Welcome to what is the third in our seminar series, Translational Research in Bacteriophage Therapy. This is, again, a series that's both sponsored by the Viral Information Institute at San Diego State University, as well as the IPATH, which is the Center for Innovative Phage Therapeutics and Applications at UCSD. Our goals, again, are to bring together groups of individuals who are interested in basics of phage biology, mechanisms of phages, as well as those who are interested in phage therapy. So let me introduce our speaker today, coming from the University of Pittsburgh, and that is Dr. Graham Hatfull. So Dr. Hatfull comes to us from Kent, originally in Southeast England. He received his BS degree at Westfield College at the University of London. He received his PhD at the University of Edinburgh in Scotland, and he's performed two separate postdoctoral fellowships. One was at Yale with Dr. Nigel Grindley, and the second was at the Medical Research Council with Dr. Fred Sanger. He's been a member of the faculty at University of Pittsburgh since 1988. And Dr. Hatfull has really been a pioneer in bacteriophage research. He's got over 200 publications from his laboratory looking specifically at bacteriophage research. And his group really has focused on mycobacteriophages. Dr. Hatfull also worked to establish the Phage Hunters Advancing Research and Education, which is called the FIRE program. And what most people here have probably heard of, he's also worked to establish the Science Education Alliance Phage Hunters Advancing Genomics and Evolutionary Science, which is the C-Phages program. And the C-Phages program has been at 73 different institutions and has trained over 4,800 undergraduates to this point. And their goal is really to increase the interest of undergraduates in biological sciences and, of course, viral discovery as well. Dr. Hatfull is also funded by the Howard Hughes Medical Institute. And through the Howard Hughes Medical Institute, they were able to administer the C-Phages program. He runs a sizable research laboratory at the University of Pittsburgh. Again, they focus on mycobacteriophages, and they've isolated over 15,000 mycobacteriophages to this point. They sequenced and characterized about 1,800 of those phages to this point. And recently, his laboratory has been working on mechanisms by which phages and the bacterial host sort of collude together to prevent further phage infections within those organisms. And I know he's going to talk a little bit about that. He's also going to talk about some recent work where his laboratory has moved, where they're collaborating with clinicians really across the country and actually across the world in terms of helping them develop phages for bacteriophage therapy. In particular, this recent case, which has got a lot of notoriety in the news today, which I know he's going to talk a little bit about, where they were able to treat a patient with disseminated Mycobacterium abscessus with some modified bacteriophages. And that study was recently published in the journal Nature Medicine. So the title of his seminar is Mycobacteriophages, Discovery, Dynamics, Conclusions, and Therapy. So please join me in welcoming Dr. Hatfull.
Are we on? Excellent. Well, thank you so much for the terrific and very kind introduction. It's an absolute delight to be here, especially as we've had these active collaborations over the past year or so with Chip and Connie and Stephanie, and it's a special treat to be here to visit and meet with new colleagues and some not so new that I've known for a while. So terrific to be here. I'm going to talk mostly today about the instance of phage therapy that we've been involved with together with Chip and collaborators here as well as in London. And I'm going to spend much of the time talking about that. I thought that was a topic that might interest some of you. And as we go through, I'll try to say a little bit more about our efforts in bacteriophage discovery and genomics that David mentioned. And time permitting, at the end, I'll talk a little bit more about these other things that we're doing in the lab in terms of sort of the microbial dynamics and the collusion between the phages and their hosts. And hopefully we'll try to have that make sense in terms of some of the challenges and opportunities perhaps for expanding the therapeutic activities. Please stop me with questions or queries at any point during the talk. I'd be more than happy to answer them. You need to use the microphone, put your hand up, and then the microphone's at the back there, and I'd be happy to address questions. So, in early October 2017, I received an email from a colleague of mine, James Soothill, who was in London at the Great Ormond Street Hospital. I had met James about 20 years earlier at a small phage therapy conference in Tbilisi. And James had remembered that we had an interest in bacteriophages that infect mycobacterial hosts. And this was James's email message. It says, Graham, we have 2 patients with M. abscessus infections, which are proving hard to treat. One of the parents inquired about phage therapy, most likely because she had read events from here and elsewhere that brought that to attention. I imagine there would be considerable barriers to administering a phage, but if things get desperate, there could be a situation which I would regard it as appropriate and ethical. If you were happy, we could send you the strains so that you could test them. Last time you sent me the phages was a bureaucratic nightmare at this end. And please let me know if you are happy to receive the strains. So a sort of a hypothetical request to see if we would be interested in looking at phages that might infect these two patients. The patients had similar profiles. They were kids. They both had cystic fibrosis. They both had a double lung transplant. And they both had now infections with Mycobacterium abscessus, which likely pre-existed the transplantation, but then following the transplantation and the administration of immunosuppressive drugs, these had become wider spread and disseminated infections and clearly sort of life-threatening. We received these strains in early November. We dubbed them our own designations as GD01 and GD02. Both were highly antibiotic resistant, and essentially the patients had run out of options and were being treated essentially at the level of palliative care. And so just as illustration, this shows one of the patients at the point when they were suffering from these infections, and this is the sternal wound from the transplant, which was quite badly infected, and it disseminated to form these skin nodules on the limbs quite prominently. And I think what was sort of particularly scary was this infection in the porta hepatis and the liver, which was quite severe and large, and I think was threatening. And there had been episodes of essentially complete liver failure in this patient. This is all notwithstanding this long list of antibiotics and immunosuppressive drugs that this patient had been taking apparently without the ability to control this infection. So our goal was to simply take these strains, grow them up, and see if we could find what we thought we would need as 3 phages that could be used to infect and perhaps kill and perhaps control these infections. The 2 strains, GD01 and GD02, as we'll see, were different to each other. They're both abscessus, but different clinical isolates. The rationale for wanting 3 phages was simply because we feared that if we had 1, you'd get resistance to that phage, and that you'd get treatment failure, even if you had some initial success. So that was the rationale for wanting to use 3. We don't know much about patterns of resistance to these phages, and so the goal was to try to get 3 phages which are genomically distinct to each other in the rationale that there is the greater likelihood that there would be difference or orthogonality in terms of the resistance profiles. The phages should be lytic so that they actually kill the bacteria when they infect them and not temperate, because for temperate phages, there's a relatively high level of survival, which seemed to us to be highly deleterious if you want to use the phages therapeutically to kill the bacteria. And there should be other desirable parameters. You should be able to grow them to high titer. They should be stable and sort of easy to grow and to manage. So those are our goals, and that's what we were after. We figured that there are three main approaches that we'd use to try to find the phages for these strains. The last one here is simply to do de novo phage isolation from environmental samples or any samples that we could collect. The second was to screen individual isolates of mycobacteriophages that we have in our collection at Pittsburgh, and I'll tell you more about what that is and where that came from. And then we could also use strategies to essentially take pools of phages from this collection and use the mycobacterium strains to enrich for anything in there which is likely to work effectively against these strains. So we had several strategies, and at various points we've actually used all of those to varying degrees of success. And so we were doing this towards the end of 2017 and into the early part of 2018. And at least some of the bad news was that it's actually quite hard to find the phages using any strategies for these Mycobacterium abscessus strains. So we could see early on that this was going to be a challenge. For a couple of months, we tried really hard to get a phage for strain GD02. And we were sort of rather delighted when we had success at some point during January, and we had this thing we could show we could isolate and prep it up. So we had at least one phage that worked quite well, but it was too little too late because that patient died on January 3rd from the M. abscessus infection. And we essentially sort of put on hold any desperate efforts to try to get phages for GD02. The second patient, though, we had success with, and so that's the story that I'll tell you about. And so I'm going to digress to tell you about where this collection of phages came from that we were able to screen in order to find things that were the most effective and useful therapeutically. So since 2002, we've been developing what we would think of as integrated research education programs. There's dual goals. One goal is to promote and to advance a particular question, scientific question about phage diversity and phage genomics. And secondly, to promote engagement of students, undergraduate students, in their own education in science. And the two programs are, first of all, this Phage Hunters Integrating Research and Education. We love our silly acronyms. And then the second is the Science Education Alliance, Phage Hunters Advancing Genomics and Evolutionary Science, or CPhages. And essentially the FIRE program was developed as a local program for students in Pittsburgh, high school students, undergraduate students. CPhages is really targeted towards first year and some second year undergraduate students. And the CPhages program started in 2008. Currently, I think last year, about 130 institutions that participate in that program. And last year, about 5,000. So the CPhages program since 2008, there's probably been about 25,000 or so students that have gone through the program, all engaging in the same type of platform. We would think of it as a platform in which students go to and take environmental samples from their backyard or soil, compost, anywhere where students choose to go and take a sample, and they can be quite imaginative. And then to incubate with a strain of your choice, and we've used several, but most extensively with a particular strain of Mycobacterium smegmatis. And then to purify the plaques, to amplify them, to grow up phage stocks, to look at them in the electron microscope, to isolate DNA, sequence the genomes, and then annotate the genomes and do the computational biology to identify the genes and to do the sequence comparison and essentially learn about bacteriophage and viral diversity, and to try to gain insights into the evolutionary mechanisms that generate these highly diverse populations of bacteriophages. And so we've implemented this in various different formats. The Phages program is delivered as a 2-term course for freshman undergraduates, typically sort of 4 hours a week, divided in this way, and typically over 2 terms or the equivalent in a quarter system. And then we've also delivered it as an intense 2-week workshop in Durban, in South Africa, as part of the KRIT program there at the University of KwaZulu-Natal. Again, with the idea of getting students interested in doing research by engaging in and doing viral discovery and genomic characterization. So because phages are specific for their hosts, we had to decide which host to focus on. We've been working on Mycobacterium smegmatis for a long time as a model for understanding phages that infect pathogens such as Mycobacterium tuberculosis, and of course, other mycobacterial pathogens. And in fact, the program in general has focused on the hosts within the phylum of the Actinobacteria, and I've just sort of highlighted a few here that we have phages isolated on. And even though numerically the main production has been on Mycobacterium smegmatis, we have others on these various other strains, and clearly this is a fairly rich and diverse phylum of bacteria that we want to continue to explore. So the Phages program is really built around this idea of an inclusive research education community. And the reason why it works, and it works at scale with such a large number of students, is that there is a well-defined programmatic structure that provides the sort of the common elements, the training workshops, the protocols, the databases, the assessment tools, all of those parts that then individual schools can essentially plug into and do the nuts and bolts of the phage isolation discovery. And because of this sort of plug and play type of structure, this isn't restricted just to like R1 universities. There can be R1 universities, and UC San Diego is a long-standing member of the Phages program. But community colleges and tribal colleges that don't have robust research infrastructures can essentially participate in the program, and those students can isolate their own phages and name them and characterize them and contribute to this larger and this broader goal. I'm only going to show the one slide and just say very briefly that it's very clear from detailed science education sort of assessment approaches that this does in fact have a really big impact on students that engage in the program in terms of getting interested and continuing in their science education, especially compared with your standard introductory biology course, which bores the tears out of everybody. And most importantly, it really is inclusive. And so you can measure similar types of gains for underrepresented minority students, for first generation students, males, females. We see these large gains in essentially all of the components that we look at. And so we think that this is an important model for engaging students and enhancing persistence in science education. But scientifically, it's really productive as well. And so this is just an example of the number of sequenced actinobacteriophages over a period of time. I labored long and hard to sequence the very first mycobacteriophage starting in 1988 that got published in 1993. And I thought this was the only one that would be characterized to this level of detail at that time. And now it's not even a blip on this graph, which is sort of rather distressing. But nonetheless, you can see this quite impressive increase in the number of sequenced genomes. Clearly, this is in part and in large part because of the advances in DNA sequencing technology in terms of the cost and the simplicity of doing it. But it doesn't matter how cheap and simple and easy it is if you don't have the entities that you're going to sequence, right? And so the program uses this this broad inclusion of students to isolate all these phages that can be carried in this way. And so you can see the impact of the programs, I think, as associated with this increase in the number of phages. And so, not surprisingly, the vast majority of these have come from within the US. There's a few from elsewhere across the world, although we would like to expand that if we can. And a pretty broad distribution across the US where all of these phages have come from, including, of course, in San Diego because of the program here in the C phages. A critical part of the program is having a database and a website in which students can enter all of the information about their particular phages that they've isolated, and also be able to access all of the information that is collected there. So we have what's proved to be a very useful and robust database and website at phagesdb.org. It's publicly available, so you can go in there and look at your favorite bacteriophages. The students name all of the phages that they isolate, and so they have some extraordinary, sometimes amusing, sometimes rude, but we veto those. And you can see just a few of the interesting kind of names down here just that happen to be on the site. And the total collection now is about 15,500 phages. And we've completely sequenced almost 3,000 of those. And these are from about a dozen different hosts, but as I mentioned previously, the vast bulk are from the mycobacteria. And there's about almost 1,800 sequenced mycobacteriophages and about 10,000 individual isolates. And what's nice about this is that we have quite a lot of data and genomic data that sits in our computers, but we also have these archives of phages that sit in the freezer. And so then what's nice about that is that each of those are isolates that can be grown, propagated, characterized, manipulated, perhaps even used therapeutically, right? And we have more in the freezer, and we know where to go find things in there. So I mentioned that the mycobacteria, which are the group that we're really going to focus on here today, are mostly grown on Mycobacterium smegmatis in one particular strain called MC²155, which was a strain developed by my colleague Bill Jacobs, who works at the Albert Einstein College of Medicine, which explains his MC² designation. And so it's about 10,000 phages and just shy of 1,800 in total that we've sequenced. And what we've learned about the diversity and the variation amongst those phages is that there's a relatively large number of different genomic types, where we'll just use that term rather broadly. So if two phages are different at the nucleotide sequence level, we put those into different groups. We call them clusters. And so we can then assemble everything into a group of clusters, and also things we call singletons, which is a cluster with one member, essentially, right? And so one phage with no other relatives. So at this point in time, with about almost 1,800 sequenced, there's about 40 different genomic types. And within each of those types, there's a high degree of variation. So this is just to give you a flavor of what that diversity looks like, which is pretty remarkable, given that these all infect exactly the same strain of one species of bacteria. And in fact, there's only 2 instances of phages of exactly the same phages being isolated twice at different places and times. Everything else is different to each other, but they're different in every imaginable way possible. They may differ by a bunch of SNPs. They may have no SNPs, but one has an extra gene, or lost a gene, or 2. Or they may share a subset of their genes, or a relatively large number of genes, or any range of a number of genes that share between. So this is a crude and we think useful, at least to us it's a useful way of categorizing these groups. But they're really sort of bumps on a continuum, a continual spectrum of diversity of of these mycobacteriophages. And you can illustrate this, in different ways. This is a network phylogeny, essentially, that's based upon the proportion of shared genes, and these spit out what are the same clusters as labeled here: cluster A, cluster J, E, L, etc. The phages are at the tips of each of these branches, and you can see, for example, that A is very large, but it's also highly diverse with different subtypes and subgroups that sort of populate this diversity. So the variation diversity is complex, and we can at least simplify it in the way that we think about it by dividing it up into these sort of quasi-taxonomic groups. More than 50% of the genome and the genome types are temperate. Let me just take a moment to clarify what I mean by temperate phages and lytic phages. The terms are used and defined differently by different people, so I'll just try to provide some clarity to what I'm going to say. So some phages are lytic or obligatorily lytic, such that when that phage infects its host, there's only a single outcome. It infects the cells, it takes over the cell, it replicates, it makes more phage progeny, and the cells lyse and are dead. And so lytic phages typically form plaques. This is showing a plaque on a lawn of bacteria, and this looks black because all of the cells have been killed through lytic infection by this lytic phage. Temperate phages do that. That's why you see a plaque. But that's not the only possible outcome of the infection. Some of the time, and the proportion varies enormously depending on the conditions, temperate phages establish a state of lysogeny in which the cell is not killed. The phage establishes itself by the prophage, the phage genome, existing in the cell, not always, but typically integrated into the host chromosome. And these lysogens are perfectly stable and grow perfectly well, and they survive the infection, and that's why you see these turbid plaques, because there's lysogenic cells growing in the middle. And so there's nothing weird about being temperate. Most phages, arguably, in the environment are temperate, and arguably most bacterial genomes are lysogens, because you can see prophages integrated into their genomes. They may have more than one. Some have 20% of their genome as prophages, and there's probably a substantial segments of bacterial genomes that are decayed parts of prophages as well. So this is common. It's not the unusual state. It's the common state for many bacteria and many phages. And so if we just look at some mycobacteriophages, just showing some plaque assays, this is just dilutions of individual phages that have been plated on Mycobacterium smegmatis. You can see here many of them form turbid plaques because they're temperate. There's a couple here with TM4 and D29 that form perfectly clear plaques. The distinction's pretty obvious. And you would be correct in saying that D29 and TM4 are lytic phages. But what they actually are simply mutants of temperate parents which have lost the ability to form lysogens. Okay, so you have to be very cautious about interpreting the underlying type or genomic features of a phage vis-a-vis lytic versus temperate just by looking at plaques. Okay. So the question was whether there's temperate parents on this slide. For example, adiphagia and TM4 are both members of cluster K, so they're genomically related to each other. And we have sufficiently large data sets that we can find relatively close matches. Adiphagia may not be the closest match to TM4. I don't remember what it is. Might be Zoe J, which I'm going to talk about in a little bit. where you can align them and you can see exactly where the deletion happened that removed the genes that are required for lysogeny in TM4. And the same thing is true for D29 as well. Okay, so that's our phage collection that we have. And the question we're after is, can we find phages in there that infect these M. abscessus strains? And we're not going to screen all 10,000. We don't have the capacity to do that. But we can target it to quote unquote representative examples of different cluster types. And we can focus on specific groups that we know are more likely to infect other strains of mycobacteria. For example, we know from prior work that certain subclusters infect TB and are likely to have a broader host range. So we can look broadly across this set by using representatives, but then we can hone in and focus on the subtypes that are likely to be the most productive to give us candidates. Should emphasize that the de novo isolation using environmental samples turned out to be quite unproductive. Perhaps because we're not looking in the right place, but we looked at like 100 samples and got one phager. And that one phage is clearly well within the genomic subset that we already have on smegmatis. So we didn't actually learn anything very new from that, although we did get a couple of things like down the road that we haven't yet used therapeutically, but we might use them. But this screening of the excellent collection so far has been the most productive route to identifying the phages of interest. Yeah. So when doing it by enrichment and isolating on smegmatis, I should repeat the question. The question was, what does this frequency of recovery compare with the C-phages program using smegmatis? Using direct isolation, so just taking the sample and plating right on the strain, about 10% of samples normally give you something. By doing the enrichment, which we did here, it's between 30 and 50%. And so this is quite a bit lower than the similar approaches that we've used for smegmatis. We don't know why. It's reported there's plenty of mycobacteria in your shower heads. And so we're about to engage on an extensive program to see what we can recover from shower heads, but we don't have data for that yet. So we took that subset and we screened. And the bottom line was we had success, but it was limited success. And I'll just show here, here's the M. abscessus strain. These are just dilutions of phages of various different types. And the one good hit was muddy. And here's muddy. It infects with an efficiency of plating of 1, and it actually kills this strain quite nicely. This is a strain came from South Africa from our workshops there. And that was, it took us still several weeks to get to this point, but nonetheless, we had number 1 in the bag, and so that was the good news. Where were we going to get number 2 and number 3 from? And so, we had screened all of these phages of different types and subtypes, and you don't need to look at all the details there. You just need to see it's a long list. But we saw a number of examples where that look a little bit like this. Okay, so here's M. abscessus GD01, here's Zoe J. This forms plaques nicely on smegmatis, and it sort of infects, and it's not very impressive. And I don't think that you'd want to use this thing therapeutically because it doesn't seem to really be doing very much. It's infecting a little bit, but it doesn't have this muddy-like efficient infection. So what are we going to do about it? Well, it turns out that we had in the lab, we had constructed an engineered derivative in which we had taken this phage, which is temperate, and we engineered out the repressor gene, because we know the repressor gene is required for lysogeny. And so we engineered out a removal of the repressor gene with the hope that we could convert it from a temperate phage into a lytic phage, just as that TM4 and D29 pictures I showed you. It's the same idea, but we could do it in a defined engineering way. And the bottom line is that worked really well. And so we took this derivative, which we deleted the repressor gene, and now it infects and actually kills quite nicely the Mycobacterium abscessus strain. And that essentially was how we got number 2. I would just emphasize, I'm not going to go into the details and the weeds here, but the way we did the engineering was by using a tool that we had described and developed 10 years or so previously called BREAD, or Bacteriophage recombinating on electroporated DNA. So the engineering is done in such a way where we take cells where we've really souped up their ability to do homologous recombination, incidentally using phage genes to do that. And then we co-electroporate genomic DNA plus a small synthetic DNA substrate that, if you like, has the mutation in it. And then we recover plaques. And this recombination, essentially recombining this substrate with the genomic DNA, happens so efficiently that you can simply pick plaques, screen by PCR, and find products and sort of, purify them that now have a clean, for example, a clean deletion of the gene. So this happens efficiently in a well-defined way without the requirement for selection or for adding any genes in order to identify the particular recombinants. Number 2. Number 3 came from a related strategy, but it required two things for us to do to it. And this is a phage called BPS. We had, because we spend most of our time in the lab kind of fiddling around and trying to understand how phages work, we had spent considerable time in finding out how it establishes lysogeny, and discovered it has a new way of doing it, which is very different from the Lambda textbook model, and it uses what we call integration-dependent bacteriophage immunity. The point being that we had sort of a knowledge of, we had a basis of knowledge that we could use in order to rationally engineer these phages. And so we used a derivative similarly to Zoe-J, where we deleted part of the repressor, so we converted it from temperate to lytic. It turns out that that's not enough, though, because it infects, in terms of efficiency, infects M. abscessus very poorly. But if you go in here and pick those plaques from the M. abscessus strain and then regrow them on smegmatis and abscessus strain, and then recover and actually amplify and grow a stock on smegmatis, we now have a mutant that, instead of infecting very poorly, now infects efficiently. And so we've converted this lousy-looking thing into something that works really quite well. So the two approaches were engineering to make it lytic, and then generating a mutant, actually a natural variant, which has now gained the ability to infect that strain. Bingo number 3. And that's how we got our 3 phages. So here's our 3 phages. They're all cyphoviruses. We could prep them to reasonably high titer using cesium chloride equilibrium density gradient centrifugation. So we'd run them through them twice to get them squeaky clean so that they could be used therapeutically. And these really do nicely kill the M. abscessus GD01 strain. So this is just a matrix where we've set up a mixture of different numbers of bacteria and different numbers of phage. We incubate them, and then we simply plate out to look for survivors. And this is muddy alone Zoe-J BPS. You can see that at high bacterial concentration, there is some survival from the BPS. A little bit here at the very low end of the phage concentration for Zoe-J. Muddy has wiped everything out, and then together, they really nicely kill at every range of phage and cell combinations. So that was our 3-phage cocktail that we decided to go with. We could make these stocks at high titer and then dilute out to a cocktail at 10 to the 11 PFU. We showed that it was stable and when stored in that way. There's no LPS in the mycobacteria, so we tested to show that LPS levels were not detectable, but that was true. They were not detectable, which is a good thing. And it also meant that we were able to go in at relatively high titer, so we could therefore have a scheme to go in at a dose of 10 to the 9 PFU per dose twice daily IV, and also then to apply topically to the sternal wound and to the skin nodules as well. I'm not going to go through a lot of the regulatory system that we went through, but it was fairly substantial going through the UK regulatory system in order to get approval. And the people in London were spectacular at moving that through, although it was not quick. And most importantly, because even though these were engineered phages, they were not recombinant. We hadn't added any genes because the engineering is efficient, right? And so we can identify clean engineered deletions without adding any genes. And because of that, they were designated as non-GMO and therefore did not come under the quite strict European regulations for use of GMO organisms. Otherwise, we would not have been able to use them. And the patient would not be with us, I think is quite clear. So let me just give you a quick summary of the outcomes, and then I'll show you some of the supporting data. So first of all, we gave phage. There was no adverse reactions. There was a little bit of sweating, I think, in the first day, but that was it. At least from some patient samples, we were able to recover bacteriophages as by plating and also by PCR, suggesting at least consistent with in vivo replication of the phages, indicating the phages were getting to their targets and were replicating. Lung function improved. She gained weight. Within 6 weeks, the PET scan showed the liver infection was gone. There was no sign of it. There was closure and healing of the sternal wound. Incomplete. So the skin nodules are not completely gone. There's a couple remaining, but they have resolved really quite remarkably, but slowly. We saw no neutralizing antibodies that we could tell, although some weak reactions on a Western to maybe some of the phage proteins. And although some samples were recovered after treatment, we've not seen resistance to any of the 3 phages in any samples that have been recovered. And essentially, the patient has returned to a normal routine and is really remarkably healthy, which is sort of quite striking given that the patients who have had similar conditions have basically all died. So just very quickly, so here's some samples that sporadically show phage from different samples. Here's the PET scan showing the liver infection, and it is completely cleared within 3 weeks. There's pretty good resolution of the sternal wound, and the skin nodules have improved quite substantially. And then actually, this is the patient and her mum, and this is Helen Spencer, who I have to give enormous credit for, who's the doctor that oversaw this particular case and was brave enough to believe the promises of some nerdy phage biologists in Pittsburgh in order to do this. So very quickly, I have 5 minutes. I just want to emphasize the fact that even though this was, I think, a remarkable intervention with phages for a patient where, because we knew that the phages could efficiently infect and kill that patient, really optimize the chances that we could have a successful outcome, and we did. The question is, can we apply this to other cases of M. abscessus infections? And the simple answer is no. And it's an issue that we need to work on. And the reason is because when you take individual clinical isolates of M. abscessus, they vary enormously in the susceptibility to the phages that we test on them. And this is just showing several other clinical isolates and a lab strain. And here's some of our favorite phages. And the infection is either sort of is miserable or doesn't exist at all on these other strains. It's clearly not a cocktail that could be used for other patients, at least with these infections. And so we're trying to understand that. And it feeds into a general question that we're interested in the lab, which is about bacterial host phage dynamics and where diversity comes from in the phage population. And the model is not that complicated. It's simply the idea that if we take a host, here's smegmatis, and here's 2 phages, if you like, of different clusters, so they're genomically different. They both infect the same strain. How can they be so different and have not exchanged all of their DNA? The idea is that because bacteria have a strong selection to become resistant to phage infection, the phages have to co-evolve, in part by moving to different hosts. So phages move across the landscape from host to host at relatively high frequencies. at each time sampling different parts of the large common gene pool. And so that's by this migration of phages across the host, driven by resistance to phage infection and coevolution, is how the population gets formed. And so this is a story that you're familiar with. Here's a bacterium under assault from the phages. There's a strong selection for resistance, and this can be through changes in the receptor or by things like restriction, CRISPR, other mechanisms that defend against phage attack. And here's another bacterium. This particular phage now has evolved to switch to a new bacterium and thus to avoid these types of events. And perhaps phages can counter these by having anti-restriction or anti-CRISPRs as ways of then co-evolving and expanding their host range. What's less obvious and less clear, but I think generally true and probably broadly applicable, is that bacteriophages collude with their bacteria in order to fight against infection by other phages, other viral attack. And how it happens is that temperate phages form lysogens, and here's the prophage, and the prophage can confer things like the repressor that we talked about that protects this lysogen from being infected by itself, but it also expresses other genes like restriction and all sorts of other things that prevent other phages which are unrelated from attacking. So in other words, the defense systems come from the phages themselves, and they're heterotypic. So this prophage can specifically prevent some other unrelated phage from infecting, and therefore protect the cell through this collusion from that infection. And we think that that's relatively common and rather underappreciated. To show that this is true, you need to have a large collection of phages that all infect the same host, a lot of which are temperate, so you can form lysogens, and be able to do the kind of experiment where you make a lysogen. Here's a lysogen with a phage called SPASH, and here's a non-lysogen, and here's a control phage, which is how most are, and it plates on both. Here's SPASH itself, so this is superinfection immunity. This is the repressor that stops this. But now SPASH has the ability to prevent Crossroads and Giles from infecting. And it's a big effect. It's not a tenfold effect. It's 10 to the 5 or 10 to the 6-fold defense against that attack. And in fact, SPASH, we know, has at least 2 different systems, one of which defends against Crossroads and one against Giles. And it may have more. We know there's many genes that are expressed from the prophage, and there's likely to be other defense systems as well. And one of the things that's really sort of surprising about this, and I don't understand it, is the exquisite specificity that we often see. So I told you that SPASH defends against Crossroads. But now Crossroads is one of a set of related phages. The purple just shows genomic sequence similarity at the nucleotide level. So there's a bunch of phages here that are very similar to each other. SPASH defends against Crossroads, but it doesn't defend against any of these others. It is exquisitely specific for Crossroads. And so this is important because this behavior, this is what the M. abscessus strains look like that I showed you, right? And it raises the possibility that the reason why many of our phages don't infect particular M. abscessus strains is they could be carrying their own native prophages, not SPASH, but whatever's sitting in the genome that is preventing the other phages from infecting that we would like to get in there to kill them. Okay? And the fact that it's so specific means that you've really got to look more broadly at your sets of bacteriophages, because you could easily look at the wrong one, and if you just looked at some others, you might find the ones that work. So we don't understand from the ecological point of view why you'd have such exquisite specificity. But this is not the first time that we've seen it. And most of these systems, we don't know what the mechanism is that mediates this collusion, at least in some cases. So I showed a SPASH example. We think it's actually related to our good old friend, the prototype E. coli phage lambda, which has this Rex-A, Rex-B system. And we think that the two genes involved in SPASH are similar and perhaps act in a form of essentially killing the cell during infection by membrane depolarization. And that's a model that we can test, and we're doing. Just as phages carry anti-CRISPR and anti-restriction genes, they also can carry anti-phage defense systems. And so here's an additional level of complexity which determines why some phages infect some strains and not others. And so in this case, Tweedy is subject to defense by some phages, but it has this counter-defense gene, 54. 54 is a funny structure. It's got unique sequence at the N-terminal part of the protein and the C-terminal part of the protein, flanked by a tetrapeptide repeat. And what Tweedy does is it simply changes the number of repeats. And when it finds the right number, it completely counteracts the defense system that's trying to fight it off. So we don't know how this works either, but clearly the phages, temperate phages, are rich with defense mechanisms, and the phages during lytic growth have ways of countering that in order to determine whether or not they can infect. So if you want to understand why some phages infect some strains and not others, it is incredibly complicated. And the point is you really need to understand this if you want to convert a personalized therapeutic into one that you can use more broadly against a lot of patients which have a lot of variants of these types of infected strains. I'm out of time, so I'm not going to say much about TB, but I want to say that I think TB is potentially a good and an interesting target for phage therapy. So we're very interested in that and hope to be able to pursue that in the not too distant future. So this was a good day. I have lots of people to thank for doing this. So Becca, Dedrick, and Carlos Guerrero were the key people that did all of the phage characterization in my lab, and Debbie Jacobs-Serra actually did the preps for the phages that were used therapeutically. Becca and Dan did a lot of the genomic characterization of both the strains and the phages. And let me just say a very big thank you to Chip. Chip was an absolute integral part of this. There was no way we could have done any of this without Chip, because without Chip, this was a hypothetical scenario, and Chip converted it from a hypothetical scenario into a roadmap for therapy, and it was incredibly important. Could not have been done without Chip. But also thank Connie Benson for her help and advice, especially as we begin to think about TB, and of course Stephanie for all of her support and advice in going through this process. Support from HHMI and NIH, and many thousands of CPhages students and their faculty. Without this collection of phages, it's hard to see how you move forward with this. And so even though therapy was not the goal for why they got engaged in doing this science, it was clearly an important outcome. So I'll stop and happy to answer any questions.